Without further ado, let me introduce our first speaker for today, Jeff Schoenau, who will be discussing soil amendments. Jeff is a professor and the Ministry of Agriculture Strategic Research Program Chair in Soil Nutrient Management at the University of Saskatchewan. So good morning, Jeff, and welcome.
Great. Thanks a lot, Matthew, and big hello to the audience out there today in Alberta, in the Lethbridge area. Really happy to spend a bit of time here to talk to you a little bit about cropping and amendments, looking towards improving the health of prairie soils and sharing a little bit of our research results into this endeavor of how different amendments can improve the health and productivity of our soils. See if I can move things ahead here for some reason. I'm stalled. There we are. So I guess we think about how different management practices, the things we do, how we manage our soils, manage our crops, combined with amendments can improve the health, the productivity, and fertility of our prairie soils. And I think that's a very important goal towards the sustainability of our systems. And we think about what contributes to healthy soils and soil health. And I know that Yamily's going to spend a fair bit of time talking about the indicators, so I don't want to spend too much time on this, but we do have different attributes of soil. We have physical properties like soil compaction or density that does relate to things like water infiltration, the structure, the stability of the aggregates in the soil, very important. We have different chemical factors, pH, salinity. I'm going to be talking a little bit about that in more detail, available nutrients, and then down at the bottom there are the biological components of the soil that we think about related to health. Potentially mineralizable nitrogen, the ability of the soil to supply nitrogen, and the microorganisms, of course, very important in that, the respiration that they carry out. And in the middle there, you can see organic matter. And I guess when it comes to soil quality, soil health, certainly soil organic matter is very much the key, very important here in the prairie soils. And so in my presentation, I do pay particular attention to the impact of those amendments on soil organic matter, because it really is so critical to the health and the functioning of our soils. So thinking about, can we improve the health and productivity of soils here in the prairies? And maybe we think a little bit about problem soils. And one of the problem soils that we typically encounter in Western Canada are salt affected or saline soils. And indeed, there's been estimated to be as many as 3 million hectares of soils in Western Canada that are affected to some degree by salinity. So how might we address this? And thinking about this and different approaches and some of the research work that we've been doing, can we grow salt-tolerant plants, salt-tolerant forages on these salt-affected soils combined with different amendments? And when we talk about salt-affected soils, we're really thinking about soils that have a high content of soluble salts that hold back water from the plant and certainly restrict the ability of that plant to grow. And if we can get something in there that will grow and maybe combine that with some different amendments that may add organic matter to the soil, can we look at some types of improvements in those soils? So I'd like to talk to you a little bit about a field study that we started way back in 2017, and it continues to this day. Worked on by a couple of master's students that worked with me, Paul Rissick and Gravel Wong, looking at amendment application, different types of amendments, including leonardite, which is a precursor to coal that exists as an overburden on coal deposits, humic acid, char, and also good old solid cattle manure. That amendment application in conjunction with seeding a salt-tolerant grass. This is the grass that we seeded down was AC Saltlander. That's a green wheat grass. And we had study sites in south central Saskatchewan where we seeded this grass in combination with these amendments on both saline and non-saline field sites. And that's Paul there on the right-hand side, and also Dr. Harold Steffoon, who was the developer of the AC Saltlander. He's retired now, but he was also very much instrumental in getting this project going and the success of establishing this salt-tolerant grass. So the soils that we were working on, non-saline, the saline sites were what we would consider to be moderately saline, and certainly one of the big challenges whenever you're trying to get something to grow on those saline, salt-affected sites is the establishment. So you can see there at the beginning, yeah, we struggled a little bit getting things started. And in the fall of 2017, after our first year, as you can see there in that graph, we really didn't see much effect of the amendments there that we had that first year on the growth of the salt-tolerant grass. A pretty good growth on the non-saline site. The saline site, yeah, it struggled a little bit that first year. And we were maybe down to yields there that were maybe only 10 or 20% of what we saw on the non-saline site. Moving ahead a year, you can see there by the photo, yeah, things were still a little bit slower in growth there on that salt-affected soil there on the right-hand side compared to the non-saline site, but we were starting to get some pretty decent establishment there. Still a few weeds in there, some kochia, but we were able to get those under control through an application of fluoxapyr. And looking at the yield, the above ground biomass yield there in that spring of 2018, 2 years after we'd set these up, you can see there a couple of things. First, that we are starting to maybe see that the growth of that grass on the salt-affected saline site there indicated in the orange. It's improved. Now we're maybe around just about half of the yield that we're seeing of the grass on the non-saline site in the blue. Also a trend towards a positive effect of those amendments. Although not statistically significant at the 5% level, particularly on that non-saline site, we are maybe seeing some trend from those benefits in promoting the growth of that grass. Moving along into 2019, well, that was a pretty dry spring, but interestingly enough, now we're starting to see, as shown there in the graph on the bottom and looking at the pictures of the two sites and the data there, the yields, we're actually seeing very similar yield on the saline site compared to the non-saline site. And the amendments there in that year, we're really not seeing much difference amongst the amendments compared to our unamended control. But certainly that grass growth on that saline site has caught up to the growth on the non-saline site. And that catching up that we're seeing, I think, is very encouraging because basically what we're doing, folks, is we're getting a palatable forage to grow well on that salt-affected land. As shown there, 2021, you can see some very good grass growth on that saline soil site. So what's going on there? Actually, we were able to document that once that salt-tolerant grass got established, it actually developed a pretty extensive root system, despite the soil salinity. And once that root system established, it enabled those plants, that grass, to be able to use that shallow groundwater as a resource, and also the residual nutrients that had accumulated in that soils from years of adding fertilizer and really not having much growing there other than foxtail barley and kochia weed. And really moving ahead a couple more years there, you can see there that, boy, that grass growth looks pretty good on those saline salt-affected soils compared to the non-saline. And in fact, when we measured the yield that year in 2022, we actually saw greater grass yield on the saline site compared to the non-saline site. So we're actually getting a higher yield, which we attribute to that grass and those roots being able to tap into that shallow groundwater in that saline site and also utilize some of those unused nutrients that had accumulated. In 2023, we took a look at how the soil properties changed as a result of 6 or 7 years of growth of that salt-tolerant grass. Now, we need to look at this with caution because this is just a snapshot, folks, comparing to 1 year to another year. And salts move up and down in the soil profile in response to environmental conditions, particularly wet year versus dry year. So sometimes we can have a decrease in salinity just because the water tables drop because it was drier, and we have more downward leaching. But I think this is interesting because it does tend to agree with what we see out there in the field. In comparing 2017 versus 2023 soil salinity measurements in those plots, we see a small decrease in salinity in the non-saline plots, but they weren't really salt affected to begin with. But interestingly, in the saline site, we see a very large drop in the electrical conductivity, which is a measure of the salinity in that top 15 centimeters, that we can explain by that grass there, using that water there, lowering the water table, and basically enabling some of that salt to be leached down a little bit deeper in that soil profile. The other thing that we looked at was the soil organic carbon, the soil organic matter in that top 15 centimeters, comparing 2017 to 2023. And there, both in the non-saline and the saline in the control unamended soils, we did see an increase in the soil organic carbon concentrations in that top 15 centimeters. Also of note was that our amendments, where we have added the Leonardite at 10 tons per hectare, humic acid in the first year, and then biochar at 10 tons per hectare the second year, and then also cattle manure at 10 tons per hectare. There we see, folks, significant gains in the soil organic carbon content in that top surface layer of the soil, and that was particularly noted in the biochar treatment here, maybe reflecting the fact that char is pretty recalcitrant material, very resistant to decomposition, even compared to manure. So really saying, growing forages, adding the organic carbon containing amendments at that 10 ton per hectare rate, increase the soil organic carbon, and that's I think a contributor to the soil health. And really, I talked a little bit about roots, and roots are so very important. We measured the root biomass in the top layers of soil after 6 years of that grass growth compared to the adjacent annual cropping system. And you can see there, folks, really the benefit there of that forage overall compared to the annual crop, adding to that root biomass, much more roots that are in there that ultimately adds to the organic matter of the soil. And comparing the root biomass in the non-saline to the saline soil, very similar in that surface layer, moving down a little bit deeper down into that 40 to 60 centimeter depth, where you've got a lot of salt in that saline soil. You do see that having a negative impact on the root growth compared to the non-saline, but still able to produce and contribute to roots at that depth. So that root biomass, important in accessing nutrients, utilizing water efficiently, and also adding organic matter to that soil. I guess another factor related to soil amendments and use of soil amendments to improve soil health are there any kind of amendments out there that contain nutrients and organic matter? And we're already maybe most familiar with manure, but are there some other things out there that can be of value? And talked a little bit about these in our first study, looking at amendments made to salt-affected soil. Thinking about, can we use byproducts of bioenergy production as soil amendments? And so a study that we conducted a few years ago, and a PhD student who worked with me, Kayla Dalatabi, on this, and as well working with some folks at Lethbridge Agriculture Agri-Food Canada, Dr. Ziying Hao and Dr. Monica Benke, looking at how some byproducts of ethanol production, like thin stillage, wet stillage grains that are typically fed to animals, but sometimes may accumulate accumulate in excessive amounts. And what happens if you use these and apply these to the soil, as well as glycerol, which is a byproduct from biodiesel production, and then chars like biochars that are produced from feedstocks, waste feedstocks like oat hull, straw, flax fiber, can get bio oils out of it, some energy out of it, and also have some value as a soil amendment. So this work that we did, one of the first things that we did was we looked at direct land application of liquid thin stillage from ethanol production, and actually applied this very similar to liquid swine manure, which we knew from many years of research work, a very good source of plant available nutrients, a high content of readily available nutrients. And actually the thin stillage kind of had a similar composition to liquid manure, in the sense that there was quite a bit of plant available nutrient, easily mineralized or decomposed, released into the available form nutrients. So we did a study with this liquid thin stillage. And in comparison to an unfertilized control, we had thin stillage applied at different rates of nitrogen, low, medium, and high, compared to urea. And as you can see there, the thin stillage actually served as a very good source of available nutrient to increase the grain yield. In fact, per unit of N, it actually performed a little bit better than urea. And that was attributed to other nutrients that were contained in that thin stillage. And like with manure, we found the thin stillage worked better when it was injected into the soil compared to broadcast. So some of these types of materials that are produced and may end up not having a home for whatever reason can be important sources of plant nutrients. We also looked at glycerol, char, wet distillers grains in our studies. Some of this material is a bit of a challenge to apply out there in the field practically, but we were interested in what kind of a response that we saw from the soils and the crops growing on those amended soils. And this was from one of our field research sites. Actually, was a site on my own farm in south central Saskatchewan, and we can see there that comparing to the unamended control, there's urea. The glycerol alone actually had a negative effect because it immobilizes or ties up nitrogen when it decomposes. There's not very little or no nitrogen in glycerol. When we added some urea along with it, we got a pretty good response there. Our char there alone didn't give us much response in this study, but where we combined the char with the urea, and we've observed this in some of our other work, the char can act under conditions of high loss, especially high loss of nitrogen. The char can enhance the efficiency, the retention of the nitrogen in the soil, and therefore improve the recovery of nitrogen, and that can potentially contribute to an enhancement in yield. Our wet distillers grains were also a very good source and give us a significant increase in the yield as well. Manure, of course, talked lots about manure in the past. Manure, a very good source of plant available nutrient in the short term with the liquid manures, with the solid manures, more of a slow release source of nutrient like nitrogen, and more of a long-term impact. The soil organic matter influence from manure we recognize, and I think shown here the influence of manure on adding soil organic carbon here. And this is just the organic carbon concentration I show here, but from some of our trials here showing no manure, 15 tons per acre for 14 years, we do see an increase in that organic carbon concentration, that organic matter concentration in the soil. So manure is a great way to add nutrients and also build up the organic matter content of the soil. But like any other amendments, it takes time. And so if you go out after 3 years, 5 years, it can be hard to show a measurable difference. So that's why the long-term trials are really important, to be able to show that long-term impact. We did some studies with the glycerol. And with the glycerol, it is decomposed very fast in the soil. It does tie up some available nitrogen, but it did increase the organic carbon concentration slightly in the soil. The other thing that those amendments do is that they enhance microbial activity, which we measure as soil microbial respiration or production of carbon dioxide gas in the soil. And there we see the effect, for example, compared to the control there. We see the urea fertilizer increasing the CO2. In part, that is coming from an enhancement of microbial activity from the fertilizer itself, as well as CO2 produced from hydrolysis. But especially pronounced here is the effect of the wet distiller's grains, a big increase because you've got the carbon that you're added there, the organic material in that, along with the nutrient that stimulates that microbial growth. So those amendments stimulate microbial activity, biochemical activity, that microbial respiration, the biomass carbon and nitrogen, which we commonly measure in our studies, enzyme activity enhanced, kind of all point towards the important role that these organic matter containing amendments can do to contribute directly to stimulation of microbial activity in the soil. So these kinds of co-products have value as source of nutrients, organic matter, health improvers. Thin stillage, distillers grains, manures are effective slow-release sources of macronutrients and indeed micronutrients as well. And many of them add carbon as well. And some of them can also help retain the nutrient by virtue of their ability to add to the surface area, the charge of the soil to help retain them. Of course, any of these materials do have challenges, availability, the cost of transportation, practically how are you going to get them on, and the economics. But important to always think about how we can recycle something and benefit from the carbon and the nutrients and the material that's contained within. We have some studies going on right now where we're looking at the addition to soil of spent bentonite filtration earth from canola oil crushing, and to see what impact that has on the soil fertility, the soil conditions and productivity. Which I guess brings me to a study, and this is my last one I'll talk a little bit about, is specifically looking at adding amendments to restore the productivity of specific field zones. And this kind of ties in with precision fertilization, precision nutrient management. Eroded knolls that we see out there in the fields of our prairies, we can really see them when there isn't any residue cover. And this is one here, an old picture I had in my files of a fallow field. And you can see those eroded knolls, those white areas in the field. Those are areas that are typically deficient in nutrients and often deficient in phosphorus, occasionally micronutrients, and so could they benefit from the addition of those phosphorus and micronutrients? And they're also very low in organic matter. So what's the impact of adding manure as well on those specific locations in the field? And so a study that we undertook about 4 years ago was what we called Rebuilding the Fertility and Productivity of Eroded Knoll Soils. Dr. Ryan Hangs, a research associate, and myself, recognizing that the historical erosion that's occurred on those upper slopes by wind, water, and tillage erosion has created some pretty poor soils. Low organic matter, poor fertility and structure, not very good water relations, and I guess you might say collectively, not the greatest in soil health. And although we've been improving those kinds of soils through our conservation management practices, our no-till, they've suffered from some sins of the past. And can we address this through amendments? So that was the basic question that we set out to address in our research work. This study started in 2020 and ran to 2023. We looked at amendments in a couple of south central Saskatchewan fields, and as treatments we had phosphorus alone, zinc alone, and we had zinc as both added as sulfate and also added as zinc that was contained in a char that was actually made from tires that were produced into a char, and they actually contained a very high content of zinc. We had a copper treatment in there, a solid cattle manure alone, and also those individual treatments applied as well in combination. As our controls, we had no amendment, and we also had a treatment of topsoil replacement. And that topsoil replacement treatment that we had was our upper level control, and really came based on previous work that had been done by Dr. David Lobb and his PhD student working on it now, Curtis Cavers, looking at the effects of taking topsoil out of the depressional areas of the field and bringing it back up out of those depressions where it had eroded to over the past years by water erosion, tillage, dragging it down and bringing it back up and putting it on these eroded knolls. So basically, putting back in what had been lost and comparing that to the amendments that we were interested in. Folks, these were soils that were pretty low in available phosphorus and zinc. The knolls that we worked with, moderate copper fertility, low organic matter content is shown there, and also a pretty high pH as well. So what did we find? We added these amendments once in 2020 at the beginning of this 3-year study. We added them at fairly high rates. So our phosphorus rates were over 100 pounds of actual P2O5 per hectare that we applied in this study. That's another shot. So it was all RCBD, randomized, complete block design, replicated trials. And the knolls were big enough areas that we could actually set up entire plot areas in those field areas. And that's how we got the soil from the depressions down. And we had a depth of topsoil replacement of about 10 centimeters in our topsoil replacement, our upper level controls in this study. So what did we find? Well, we grew different crops. We grew wheat, we grew peas, we grew canola on these plots. And I think you maybe can see there some areas there that looked a little bit better. You can see, yeah, those actually are our topsoil replacement treatments. And so showing some of the yield data here, this is 2020 wheat yields in that first year on one of the sites. So just to take you through this is the yield of spring wheat. So we've got grain, we've got straw yield, and the total ones are in the hatched bars there. Here's our control where we had none of our amendments. We did add nitrogen to all of the plots, so we were removing the effect of nitrogen, but we had treatment there, our control. Then we had our individual nutrients, our phosphorus, our zinc, our copper added alone, and then in combination, and our topsoil replacement, our upper level control over here on the very right-hand side. And what you can see there is that the individual nutrients maybe gave us a little bit of a bounce improvement in yield. The phosphorus sometimes just applied alone. The copper and sometimes the zinc actually gave us a negative response applied alone because of some negative interactions. But where we had the nutrients applied in balance, the zinc to copper along with the phosphorus, and also where we had those with the cattle manure, and even the cattle manure alone gave us some of the highest yields that we saw. But oftentimes those yields, depending on the crop, could not approach what we saw there with the topsoil replacement. We had peas. Of course, peas can fix their own nitrogen, and they're pretty darn good scavengers of nutrients from the soil. There we saw a positive response of the peas to zinc fertilization because pulses can be responsive to particularly responsive to a lack of zinc and therefore to zinc fertilization. So we saw a positive response to the zinc fertilization. Where we got to some of our highest yields again, where we had the micros in combination with our manure. But because those pulses are pretty good scavengers and can do pretty well on some fairly poor soils, in this particular case shown here, not a lot different in yield from our topsoil replacement for that crop. Water relationships, interestingly, we look here at the control and our single application of cattle manure, not a lot of difference there. It takes a few years of manure addition to actually change the structure, the organic matter content of that soil significantly to improve it. And I know work by Dr. Frank Larney at Agriculture Agri-Food Canada here in Lethbridge, some pioneer work that Frank did a long time ago, just showing that, you can certainly adding manure to those eroded areas in the field can be beneficial, but it's pretty hard to get back to get all of those good things that topsoil does. And this shows here that, we really got our biggest benefit here in the water infiltration from where we actually replaced that topsoil. 2021 on our study site, well, that was kind of a write-off because that was a really, really dry year. But moving on into the next year, this is 2 years after we made our amendments. We see here with the wheat, we're getting very good response here from our combinations, our appropriate balances of micronutrients and our phosphorus together. If you just apply one nutrient alone, like phosphorus, and you don't apply any zinc, you can end up with imbalances, and actually what's commonly observed is a phosphorus-induced zinc deficiency when you apply high rates of phosphorus to a soil that's marginally deficient in zinc. And I think we're seeing that here, but where you got the right balance, yeah, good response. Also where we have the manure treatment, and here not a lot different than the topsoil in this year. And in fact, with the canola, we were actually seeing some very, very good responses 2 years after to those treatments, particularly where we had that balanced availability of nutrients, folks. 2023, one more year here. Interestingly, in this particular year, it was the topsoil replacement treatment that really showed up above all the other treatments. So it depends on the year. I'm not sure why it was on that particular eroded knoll, but we really saw benefit from that topsoil replacement. And we got some response from our other treatments, but not nearly what we saw. And I think some of these other ones, we were maybe starting to run out of nutrient in those ones, whereas in the topsoil it continued to provide all of those things that those crop plants really needed. So topsoil replacement has the longest lasting effect. So I guess in conclusion, when it comes to amendments from our study, depending on the crop, we saw some positive growth responses there. In the first year, we saw good responses to the combinations, especially starting to see those in the third year, and maybe that related to the fact that it just took a little while for those amendments to actually get down into the root zone where the roots were, but indicating some potential short and longer term benefits from those amendments in restoring productivity, but especially I think showing the importance of balanced availability, and also certainly again the ability of manure to bring into a play a lot of those things that are important to plant growth, not just available nutrients, but also improvement in the structure. But I think overall, it's pretty tough for any kind of amendment out there to really replace all of those good things that the topsoil does. So very, very important, therefore, to keep that topsoil in place to begin with by preventing that erosion from occurring. So certainly, I think if we look after our soils and crops through sound stewardship, which includes the kinds of amendments that add nutrients and organic matter, I think folks they certainly will look after us. So with that, Matthew, I will thank everybody for the opportunity to come out, and thank you, the Lethbridge County, for the opportunity to participate.
Awesome. Thanks, Jeff. That was a great presentation. Like I said before, if anybody has any questions for Jeff, throw them in the Q&A and we will get to them at the end of the webinar. We'll get right into the next presentation. Our next presenter, Yamily Zavala, is the Soil Health Lab Manager and Soil Health and Crop Management Specialist with Chinook Applied Research Association, located in Oyen, Alberta. Yamily will be presenting on soil health indicators. So good morning, Yamily, and welcome.
Thank you very much for the invitation. I just going to, Jeff, thank you very much. You did good information there. We need to talk after this. Sorry, my apologies. Okay, so I'm just going to be talking about soil health indicators and why they are so important for us to know about them. So, okay. Jeez. This is stuck here. Okay. So soil health, what are they? They represent the living essential of soil. It's the connection, the biological, chemical, and physical aspect of the soil, of which is ecology and agricultural function, how the system function is related to the soil health indicators. So it is a living system. It is not an inert material. There is a lot of activities in the soil, and they are driven by the biological component in the soil. So in order for us to understand how the soil status is, we need to sample the soil in a different way. So with that in mind, we need to look, when we do sampling, we're just looking at a way of, most of the time for fertility, for knowing how much NPK we need. But in this case, when we're talking about soil health, we just need to see the soil in a different way. There is a protocol that we have in the Cara Soil Health Lab where we can see in detail how we need to be sampling for soil health. To me, it's very important that we understand how to do the sampling, because when we sample for soil health, we need to collect the samples in a way that they are undisturbed zone of the soil. So when we do evaluation for physical assessment, we will have the soil that is going to be representative of the condition that we have. We use this sleeve. They are about 2 inches diameter. They are really, really, really hard to take the samples. I get a lot of complaints, but it's because how bad our soils are that we need to start looking at this in a way that allow us to improve it. And that's the reason when we collect the samples with a diameter of the core that we're taking, we are getting representation of the sample for us to do some of the physical evaluation, like the wet aggregation stability. Another thing that we do when we're collecting sample is that we need to measure the compaction of the soil. And the reason for this is because we are looking at the living component in the soil, which is the microbiology. And then when we measure it, the compaction of the soil, we see that we have some compaction at about the 0 to 6 inches. We need to take the samples in a way that will give us indication of what really is going on in that area, 0 to 6 inches. And then we have been recommending do samples 0 to 3 and 3 to 6 inches in order for us to see how the biology is performing in there. So that's one of the things that is very, very important when we're collecting samples. Compaction, a good amount, area representative of the soil, because we need to look undisturbed soil for us to measure the aggregation stability of the soil. So why we need to do benchmarking our soils? Well, in this case, is because when doing soil health benchmark, we're establishing a reference point which allow us to identify and document which one of those soil indicators are really functioning properly. And then also allow us to do comparison over time. And then the indicators is, as Jess was mentioned, some of the biological, chemical, and physical indicators. So why they're important? As I mentioned, we will be making track of what changes we have in the soil, and also see the variability of this based on the management that we're doing. We can see the nutrients imbalance. We can see physical constraints in the soil, and also would allow us to tailor management practices, which is going to be specific for those soil constraints that we have. And then they will allow the producer to start improving the soil. So that's why it's very important that we start building soil resilience by knowing what are the problems that we have, and we can start kind of fixing it. So this looks kind of in a different way that Jeff was presenting. But to me and what I've been learning, I've been focused on seeing that the biology component should be in a priority at this point, because of the conditions that we saw, the condition in the soil that we have been showing that one of the soil have been so messed up, sorry to say it in that way, that biology is the one that will help us, along with, all the physical and chemical, management that we can do here, improve the conditions of the soil. You can see here some of the soil health indicators that we have been put together in order for us to do this kind of evaluation. We can see the direct measurement. We can use the microscope, so we will be seeing how is in a quantitative way and by the numbers that we can generate that, but also by the diversity that we can see through the microscope. And you will be amazed what we can see there. So that's one way, and it's one of the most fascinating part when producers start looking at the soil microorganisms direct measurement, they can see the changes of the biology based on the management that they are doing. The indirect way that we measure that is through the respiration, the carbon-nitrogen ratio, the active carbon, which is that carbon that is kind of the food for the biology, and also the organic matter that we have in the soil. For the physical, we have been putting together measurements of soil aggregation stability. We need to measure texture because there are certain soil indicators are being affected by the kind of texture that we have in the soil. So we need to have that information, and you will see in a minute. And there is some measurement that need to be taken in field, because those measurements like a compaction, as I mentioned before, water infiltration, and also collect the samples in a way that allow us to see the bulk density for calculations of how much organic carbon can we start storage in our soil. And some of the chemical one, well, we need to look everything in the soil that has related to, the chemistry of the soil, the macronutrients, the micronutrients, the pH, the electric conductivity, the cation exchange capacity. All of this and much more is important for us to understand the whole picture. And one of the things that I just do an example, let's see the biological component. We are here at the top. We have too much of certain, pests in the soil that causes too much diseases. So that means that the soil is going to be at the top here. The biology is influencing the soil, and then the soil is not healthy because there is no balance in the soil with the other components. Another example will be something like, let's say that we have some compaction in the soil, or the aggregation stability of the soil is not good. So we have that at one extreme of the physical component, we're going to be having some trouble with the biology, because the biology won't have that house for them to express themselves. And the same will happen if we have too much of certain nutrients, or we have imbalance on there. We will be affecting the biology component, will be affecting the physical. So all of this, what I want to show is they are interrelated. And when we have then imbalance moving in direction to the center is when we have a healthy soil, because there is some balance, or there is some, way for the system to start working properly. I'm just going to give an example of the report so we can see how the indicators are presented here. This is a typical report where we have constraints page, which shows based on the score color, shows which one of those soil indicators is causing some trouble. And we also have a suggestion page, which shows what kind of management we can put it into there. One thing that we look at this point, I'm not concentrated in the results numbers. I just look at the score. And then that score, which is color code, will give me indication how good or how bad is the soil. When that is in red color, it's a warning signal. And that's when we need to pay attention of which one of those constraints is causing that problem. In this case, we can see that we have some physical constraint and we have some chemical constraint. So, but it's very important that we need to take into consideration that no chemical constraint is resolved if there is no improvement of the other soil properties that we have with some problems in there. This is an actual sampling from a lady from Saskatchewan. She was one of the first one that did a really good job because she measured the compaction in the soil and realized that she has this issue with compaction, and she collected samples 0 to 3, 3 to 6. I was so impressed when I received her samples, and look at the issue that she has. You can see a lot of red here, a lot of issues going on, and this is how we can start kind of monitoring what's happening in our soil. If you look at this table, this is a new table that I've been putting together to show baseline for carbon storage that we have in those soils. You can see here that all the soils that she was what we analyzed, the TOC, or the total organic carbon, which is the measurement for carbon uses for carbon sequestration monitoring, you can see that they go from 17 to 22 tons per hectare. That's the highest number that I found for her soils with all of the limitations that she has. Here is another example. So another producer that he had been categorized the soil by best and worst. Let's concentrate it in these two, and then we can see the report. You can see the difference in color. It is more blue. This is the best soil, and then it's more bluish, and the worst has a little more red. So that's an indication of how quickly we can see our report and see which one are the constraints that we had there. Now, look at the same evaluation for the organic carbon. For the best soil, they have 42 tons per acres, while for, and even having a very low bulk density, when they have, for the worst one, they have pretty much close if we add the 0 to 6 inches, because the best soil was 0 to 6 inches, and the worst was divided in two. It's pretty kind of little, we can say that is more carbon in the 0 to 6 inches for the worst one, but we can see that for the best one, we has a more depth with a lower bulk density, which is an indication that they have pretty much more organic, even more organic matter, more organic carbon into that area, which is an indication that is much better than the other one. Now, when we look at the biology of this soil through the microscope, I just want to call my attention here for you to see. So the worst soil has less protozoals at the 0 to 3, where pretty much most of the roots were growing. And one thing that I noticed here is when I look at that type of flagellated diversity that they have low count, even though they have a little more deep into the profile. Some of the counts were not as much as we found in the best soil. But another thing that caught my attention is, well, why that soil could be, worse, even though deep into the profile, they have more of these, creatures in the soil. you can look at here at the nematode population. The one that are the bad nematodes are very high. So this is an indication that in that soil that is the worst, also the nematode has an effect in probably, piercing the roots, damaging the root, causing some problems there. So this is what the report give you indication of what's really happening in the soil. This is another farmer that I wanted to show when you start monitoring your field over time, especially when you are doing some application or some amendments. This is the sampling. I've been monitoring this since 2021. This is some of the sampling. Pretty much most of the area are similar where they are. Look at the protozoas, which are this number right here. It was between 33 to 156. In the fall of 22, we went, collect a few samples, and then they were kind of, still in the low level. And what this producer was doing was some amendment application of the Johnson & Sue bioreactor extract on the seed. Only he was coating the seed with this and put it in the furrow when he was seeding. And in fall 2023 that we collected some samples, I was so amazed to see how the protozoas flagellate specific were from, let's say, 100,000 on average to millions, 5 million, 3 million. The lowest was 94 in some of this spot that we were sampling. This is an indication that when we do any management in the soil, or when we apply new activities in the soil, we can improve the biology in the soil that at the same time will improve the overall physical conditions of the soil. Another example that I have here, this is a producer that sends his samples, and then he called best, worst, and average. So you can see that the worst has a low fungal biomass comparing with the best and the average. And when we look at the protozoas, in this case, the flagellates, we see that the best has a higher levels of flagellates. Why is important this? Because think about this. The protozoas are the ones that eat bacteria. Most of them eat bacteria, amoebas, flagellates, and ciliates. And when they eat bacteria, bacteria are loaded with all those nutrients that are being taken from the decomposition of the organic matter. What happens is these protozoas take all of these nutrients that come from this bacteria, and they take what they need, and the rest is pooped out. So that release that happened there is nutrient for the plant. So this is what we call nutrient cycling as a part of that. So that's why when we see that we have certain numbers of flagellate, they correlate to get best field in this case. Another thing that Jeff mentioned a little bit about there is how important are the ratios, the balance of nutrients. This is just an idea. I'm just going to go very quickly with some report where we show that, we have all of this issue. And then I've been connected with a researcher from the state, Dr. Mir Sayed, that he had been working very close with these ratios. So I was able to learn from him and put together some ideas that I have in relation to the ratios. And this is what I've been calling this mirror nutrient balance ratios, because this shows, what could be the ideal one. And if we're able to start looking at the minerals in the soil in this way, we can start improving the soil microbiology in the soil. We can improve the soil, also physical conditions. And then there are a few of them here that shows, for example, nitrogen sulfur is supposed to be in a ratio 5 to 10 to 1. So that will help protein synthesis in microbes and plants. They also will help, in all of this nutrient cycle, which is very important, even for soil organic matter decomposition. So some of the creatures in the soils need to have, certain levels of sulfur and nutrient ratio in order for them to work, properly. There's another example, calcium-magnesium ratio also is very important because they will enhance microbial cells. It also will help in the flocculant effect of the soil, so to keep in the soil together to form more aggregates. Here are some micronutrients. Jeff mentioned about phosphorus and zinc. And then if we don't have certain ratios there, so if we have one of high minerals, it will jeopardize the uptake of the other one. So that's why it's important that we can also look the chemistry of the soil in this way. More often, this is just to show each one of these ratios has an influence in the physical, soil properties, and in the microbiologies in the soil. And also it's important that we can have then in a way that allow those system to function how properly. That's what I'm going to say. So so that's why they are important. We need to balance them. So we will enhance biological activity, and then, all the nutrient cycling, and then we'll improve the plant's resilience, and then we'll help, nutrients, and then gene potential and all of that. So that's one of the things I say, encourage beneficial microorganism activity is very important. Will enhance the plant health, as I mentioned. So if you have a plant that is healthy, they will exudate more, some exudate, which is food for the biology in the soil, and the biology will start, doing their job, kind of improving more the soil. In that sense, they will have a better house for them to live because they will have a more stable aggregation in the soil. So more of the same. So we need to nurture the soil by, improving the conditions. So this will create what we call resilient soils, and that's what we need to be working on. We need to start caring for the soil, and that we need to look at the soils in a different way. So that's why I've been presenting these soil indicators, because allow us to measure that soil in a way that we can start managing based on the changes that we can have with this soil indicator. So a benchmark is health indicators. As I mentioned, important that we need to measure it, like the functionality of the soil. And then when we have that, allow us to start management alternative for improving the soil. So what do we do? First, as I mentioned, we identify the constraint. Then we set up strategies, and we see if that strategy that we have been putting there will, go in the direction that we want. And that's why we need to go into the third option, which is monitor those changes and do certain adjustment. And then we keep, going into the third one, because that would allow us to see we go in the right direction. So the overall recommendation will be to fix first those constraints that will unlock other constraints to start healing the soil. As I mentioned, physical constraint needed to be, to me, will be the first one to start, because we need to get a better house for the biology. So the biology will do their job when we have that physical constraint, kind of eliminated. Now, remember that I mentioned the scoring curve and the color and all of that? Well, you can ask the question, where did that come from? So we in Alberta started doing a soil health benchmark that started in 2015, I believe. And then each one of the associations in the province was part of this, 11 of the 12 associations, collecting samples. And I'm not going to go into the detail, but that allowed us to get a lot of information, soils through the whole province for us to create a baseline. So if you are interested in knowing more about the benchmark, you can scan the QR code here, and then you will be able to see more in detail what was the project is, and then we can continue what we're doing right now. So there were 11 of the 12 association part of this project. So far, we have processed more than 4,000 samples, and so part of the report include benchmark and scoring system that were developed with the information that we have here. So the database that was generated, we would just took the mean and the standard deviation, and that was done based on the normal distribution for the test results that we have. And with this, we develop a scoring curve for every test, every soil health indicator that we have. And those scoring curves is what allow us to get use of results that shows where your soil fits relative to the other soils in Alberta. So that will allow you to start monitoring your soil. We have a separate scoring function for coarse, medium, and fine soils across the province for each one of all of this. Here is an example of those. So here is, you will ask, well, what are the means, If you are red, that means that you have a very low score based on the results that we have. And where this comes from is a platform that I use from Cornell University framework, that what they have the CACH, which is Comprehensive Assessment of Soil Health. And then with that information, I was able to develop our own scoring function, which every information that we have in the report is related to our conditions. Here is another example. This one was for microbial respiration. The one that you have here, this is for Alberta soils. And here you have wet aggregation stability, which is give us, information for us to calculate the score for your report. I just want to go very quickly with management that have been done in soil that has an issue, and then we can see how you can improve on your soil when you start looking at the different components that we have. In this case, if we have some issues with compaction and poor aggregation stability. This was in 2015. You can see to the top one, this is triticale that really grow very poorly. But when this was combined with others, a mixture of other crops, they were kind of helping each other. And you can see side by side how well the triticale was doing in a mix in comparison when it was growing by themselves. All of this is just to show how when we had, and that year, 2015, was a very dry year. That's when we had all of this issue that the plants didn't grow well. But when they were together, there was an interaction there that allowed the soil to be in better shape when more than one crop was growing together. So here are some examples of how the roots were start improving the aggregation of the soil. All of this shows that when you have all those plants growing together, they were able to start forming aggregates at the rhizosphere, making the soil much stable. This is another example in one field that is the beginning of that change due to the cocktail crop. So you can see soil compacted in the same plant, and the other was aggregated for the effect of the biology that was there. So this is what really is happening there. It's just the formation of the aggregate and the stability of the aggregate for the roots that are able to, and the microbiology that is in the soil. And this is exactly what we were seeing in our field, what's happening in that soil. Another example that I want to show very quickly is this one. This plant, this cocktail mix that was put there, it only grew 2 months and 10 days. And this is the surface of the soil that didn't have nothing growing there. But look how the biology with this cocktail mix was improving the surface of the soil just to be seeded for 2 months. Look at the roots again. So it's very important that we are able to make this happen in our soil so we can start producing the aggregates. So formation and stabilization of soil aggregate will contribute to the process of start the healing process of the soil. I just want to show very quickly some of the biology that we have. This is our spores that we have. So yes, we had mycorrhiza in the soil. We need to wake them up. Nematodes. These are a lot of nematodes that we have. And then we have the good ones and the bad one, that's the root feeder nematode. We have nematodes that are predatories. We have a lot of biological interaction in the soil. These are fungus that attack nematodes. This is a bad nematode. I've been seeing a pattern of the same nematode fungus in this specific nematode. Is an indication that the soil still has some biology there. Another case, this is just a trapping fungi that I was able to record, but I'm not showing here. So this is a fungus that produce on circle where the nematodes get attracted and then they constrict and they kill the nematode. This is another of those creatures that we have in the soil. This is a zoospore that attack the nematode. In this case was one of the good guys. And then I witnessed how these zoospores were attacking the nematode until it was dead. So just want to show that very quickly. And then because every soil is unique, it has its own biosignature. We need to feed those creatures and nurture them in order for us to know what's really is happening there. And what is not measured cannot be fixed. We welcome everyone to come. This is even the kids. I have a lot of fun with them. We learn, they have fun, and then we learn a lot. So this is what I have to present. And then thank you for the invitation. I hope you have lots of questions.
That was great, Yamily. Thank you so much for doing that for us.
Yes.
We do have a bunch of questions here. So, first question for you, Yamily. How do we address for the variability of the soil compaction sampler as it varies from sampler to sampler?
Well, in that case, we need to do the give me 1 second. We need to do use the penetrometer with the conditions that are more representative of the cropping season in order for us to measure it using the penetrometer, and always sampling, always measuring it when the conditions are similar. Because if you have more moisture in the soil, you're going to have kind of different, because the penetrometer will be able to. But if you select a time where you see the most representative of what's going on in the field, that's when you need to use that for.
And is there a specific brand that you recommend or that you use?
We use one that is the Dick and Johnson, I think it is. The reason we use that one is because that one was calibrated for the 200 psi and 300 psi. So the 200 psi give you 70% of the roots are able to go to 200 psi. And when you get 300 psi with that specific penetrometer, the roots doesn't go deep, it just go lateral. And then when they find, a little hole or a little moisture, they go down again. So that's the one that we use.
Awesome. Jeff, I see you answered a few questions there. We'll just bring them up here just so that I'm sure there might be a few people that have the same or similar question too. How available is biochar in terms of the quantity needed to remedy a high salinity soil, and can biochar be used or applied to adjust nutrient recommendations?
Yeah, so I guess first thing I'd say is that any amendment that you're adding to a salt-affected soil, the real source of the salinity and the problem that's not going to go away is a water problem. That you have a high water table and the water evaporates and leaves the salts behind. So any kind of amendment that adds organic matter, whether it be a char or a manure, really its main benefit is going to be, once you address the water problem, maybe establish some drainage, lower the water table, that char or that manure, by virtue of the added organic matter, is going to be able to, as Yamily talked about, improve the structure of the soil, improve the water relations, help the downward leaching, moving those salts out. But in our research work, we found that any of those amendments and the benefits that we saw actually were more pronounced in the non-saline soils. And in the saline soils, we really didn't see any benefit of those amendments, including the char, on reducing the salinity, Matthew. I think coming back to the question there about, and I'm just going to get back to it here. Okay, where are we here? Yeah, where we saw the biggest, can biochar be used to adjust nutrient recommendations? In our research work, we've seen the benefits from biochar arise when we had soils that were very highly leached, lots of water moving through the profile, so sandy soils, highly leached soils. The biochar in those instances could provide some benefit by aiding in retention of nitrogen, preventing it from being lost, and that can translate into a yield benefit. Some other chars that we worked with, I can think of one that was derived from manure as a feedstock, actually could provide some phosphorus to the soil, and also some of those biochars can also help retain the phosphorus and retain it for future years work and future years use, I should say. So yeah, I think that the salinity in itself is something that you really have to address first in relation to the water problem, and actually lowering that water table and giving those those salts a chance to be leached out. The organic amendment that you make then on top of that could be beneficial in aiding that process.
Nice. Another question we got here. So, do microbial populations vary drastically throughout the field and even throughout the season when they experience different ambient conditions? And how do you account for the variability within the field?
What we do in the field, what we do in the lab is especially for yes, there is a variability, of course. So that's the reason we recommended to do a soil health benchmark in the area that will be representative of most of the field and concentrated in that area. So when we received the samples, we practically were measuring is the potential of the biology that they have there. For example, protozoas. That's the one that the most fascinating one is. We take the samples, we keep it in the refrigerator, and then we're gonna able to measure this. We take the sample and we incubate for extracting nematodes. It's gonna give us all the, part of the nematode that is a certain amount of the soil. And also we incubate the protozoars with different dilutions. And that allow us to see, how they will perform, or how is what I call the potential of those protozoals, or what they have really there. Because when something changes in the soil, most of the biology, they got kind of dormant. They stop the activity that they have. But what we do is just we prepare some what I call soup or a broth with some soil. And then when I put the solution there, they come back to life. So that gave me an indication of what's there. Because if I take the sample and measure it directly doing my dilution, I won't find them just measuring them direct. So that's the reason the biology component for us take a little more time, because we incubate the samples at least a week or 2 weeks, depending on what we have there.
Nice. Okay. Let's see here. Jeff, how deeply did you till the topsoil amendment plots, and how much mixing of eroded knoll and topsoil occurred?
So in that, I think referring to the topsoil replacement trial, the amendments that were applied for the fertilizers were placed in bands. The manure was broadcast on the surface, and it was lightly incorporated, a shallow tillage. The topsoil amendment that we had, our upper level control, the topsoil replacement, it was basically hauled up, put onto the plots in a 10-centimeter layer, and there was really no incorporation or tillage. The crops were direct seeded right into the into the treatments after that.
Okay, and then for you, Yamily, do the ideal nutrient ratios vary much between crops?
Well, what I learned from Dr. Mir Sajed is that they have a ratio, that is this level and that level, and pretty much most of the crop are in that area, but we need to make the recommendations based on the needs of those specific nutrients of the plant, what the real crop needed based on that. And the other thing that I learned is, even though those ratios are there, we will be really hard and difficult that you can get it into the ideal one when you start doing this, because there's so many, factors happening there. So what you need to do is just start. Find a way to start and see which one of these ratios is the one that you can start, put it into the system to, again, ignite the process. And then after I squeeze Mir's brain, I put all of those together, and then I developed this spreadsheet where I can put, the numbers, and they do the calculation based on what we start playing with the different ratios to see which one could be. I don't know if that answered the question, but
yeah, I think you nailed it right on the head there. One other question for you here is, how are the soil health scoring functions established? Does it differ by crop management systems or by crop? And can I use the soil health metrics to address my nutrient recommendations in season, or it's more for the environment than yield? That's a long question.
yeah. So the scoring functions that we put it together is based on all the information, all the soils that we collected, and then we did just the the mean and the standard deviation of this. And then because the samples were collected across the whole province with the different, conditions that they have there, the one that I'm presenting there is just for the whole province. But when we look at the one that is just specific by region, depending on where the association are, they varied a little bit based on the management that they have. For example, areas that are pasture, that has less activity, like perennial, they used to have a higher weight aggregation stability than the area that had been, annual cropping system with the destroy, let's say like that, the aggregation in the soil. So based on that, that is how we developed that by the samples that we were receiving. And then we can see based on the management, which one of these, will be the one that will be more specific for that field. And they said, can I use the soil health metrics to address my nutrient recommendation in season? It's more for the recommendations that come from there for the nutrients is the chemistry. It's exactly the same chemistry that you do for your analysis. So it's just the recommendation. It will be that one. The good thing is that if you are able to see what are your, constraints or what are these minerals you are able to balance, that will be the best, because it's not just NPK. You need to look at the micronutrients, in the soil in order to not only for the plants, also for the micro. So it's all is, a holistic management. And it's the same, it's exactly the same. It's just more information.
Great. I got two final questions, and then we'll end the webinar. Are seed treatments negatively affecting the biological activity in the soil? Some farmers are claiming that they no longer are seed treating because it's killing the good things in the soil.
Yep, yes, I have a really nice example of that. So I has this, I asked my team, don't use treated seed, okay? And so they planned, because all of this was just using untreated seed. So when I went to the field to check what was happening, this is what I find out. I see there was some variability in the growth of the plants. They were right there. And then I start pulling the plants, and then I see, the one that has no treatment, the roots were having all of these aggregates starting that you saw there. But then when I pull one of the other plants that were not performing properly, guess what I find out? It was treated. So when I came back into the office and then I start questioning, guys, I told you not to use this. And then it turned out that they came to the office because they ran out of seed, and then talked to Diane, which is my manager. And then maybe Diane was busy and she said, yeah, we use that. And then they added some of this, it was a demo that I had there. They added the seed that were treated, and then immediately I could see that the plant was not performing properly. So yes, it's an indication. I know more than one producers that having found that they treat the seed bag with the Johnson and Sue bioreactor is one example, with all of this biology that the extract have, and that's why they have more biology growing in their fields.
Okay. Okay, I see there's a few more questions, but we are.
I can answer this very quickly.
Yeah, for sure.
No, Dr. Mears nutrient balance, they are no online. This is something that I've been putting together, and then I'm planning to work with him. And if there is any producers that are interested into that, we already start doing that. And then it works pretty well.
Awesome. Okay. Well, I'd like to extend my thanks to everyone who joined us today, and a special thank you to Jeff and Yamily for their informative presentations. Really appreciate you guys taking the time of your day today to present to us. Please join us again next Monday, February 10th at 11 o'clock a.m. for our next session featuring presentations on bale grazing by John Dunisveld and regenerative management by Emily Fulston. Thanks again, everyone, and see you next time.